Hey everyone, and welcome. Today we are tackling a super important topic for your CUET exam, gene regulation. Seriously, we're going to break this down so well, you'll be able to handle any question they throw at you. So you'll definitely want to stick around, because we've got some awesome practice questions and memory tricks that are a total game changer for your score. All right, let's kick things off with a big question. Just think about this for a second. Every single cell in your body, whether it's in your brain or your big toe, has the exact same set of genes. So why isn't your eyeball making stomach acid? Kind of weird, right? Well, the answer is this amazing process called gene regulation. And today, we're going to unravel that mystery and get you totally ready for your CUET. So first things first, we're going to zoom into the world of bacteria to look at this really clever system called the Lac Operon. And trust me, if you get this, you've unlocked the secret to how genes get switched on and off. This is a classic concept, you've seen it in your textbooks, and you know what? CUET's examiners love asking about it. So what exactly is this Lac Operon? Well, it's basically a perfect little model that shows us how gene regulation works in simple critters like E. coli. It was figured out by two super smart scientists, Francois Jacob and Jacques Monod, and its whole job, its entire purpose, is to let the bacteria use lactose, milk sugar, for energy. But here's the clever part. It only does this when lactose is actually around. It's all about not wasting energy. Okay, take a look here. The best way to think about the operon is like a control panel for a tiny factory. You've got a few key players. First, there's the regulatory gene I. This guy makes something called a repressor protein. Then you've got the promoter P, which is like the landing pad for the main enzyme RNA polymerase. Right next to that is the operator O. This is the actual on-off switch. And finally, you have the structural genes, Z, Y, and A. These are the blueprints for making the enzymes that will actually break down the lactose. And this is where you see that light switch idea in action. It's so cool. Look, on one side, when there's no lactose around, that repressor protein we talked about literally sits down on the operator switch. It just blocks everything. The switch is off. No enzymes are getting made. But check out the other side. When lactose does show up, it acts as something called an inducer. It grabs onto the repressor, changes its shape, and poof, the repressor falls right off the operator. Now the path is totally clear, RNA polymerase can zoom down the track, and the switch is on. The factory starts making the enzymes it needs. Now let's get super clear on this one term, the inducer. This is a really important player. In this case, it's a form of lactose called allolactose. Its job is so simple, but it's totally crucial. Think of it like the key that starts the whole engine. It binds to the repressor protein, shuts it down, and basically sends a message to the cell that says, hey, the food's here, let's get the production line going. Okay, everybody, listen up. This is a huge, massive QB trap question. I see students get this wrong all the time. They'll ask you where the inducer binds, and it's so tempting to say the operator, right? Because that's the switch. But that is wrong. The key thing to remember, and I mean burn this into your brain, is that the inducer binds to the repressor protein. Let's say it together. Inducer binds to repressor. Got it? Awesome. All right, so we've totally nailed that little switch in bacteria. Now let's zoom out. Way out. We're going from a single genetic switch to the entire instruction manual for a human being. We're diving into the Human Genome Project, which was honestly one of the biggest, most ambitious science projects ever. The goals for this project were just breathtaking. The main mission, led by a guy named Francis Collins, was to read and map out every single gene in the human body. Think about that. They had to identify all our genes, sequence all 3 billion DNA base pairs, build these huge databases to hold all the info, and, this is really important, they also had to think about the ethical side of things, what all this knowledge would mean for society. Just to give you an idea of the sheer scale of this, look at that number, 3 billion with a B, not million. Billion. That's how many chemical letters the A's, T's, C's, and G's make up the complete instruction book for a human. CUT loves to ask for specific numbers, and you absolutely positively have to remember this one. And, this project led to some incredible discoveries. Here's one of the most amazing ones. Genetically speaking, you, me, your neighbor, someone on the other side of the world, we are all 99.9% identical. Think about that. All of human diversity, everything that makes us look different, act different, It's all squeezed into that tiny, tiny 0.1% difference. It's just a wild reminder of how similar we all are. Okay, now this next fact might actually blow your mind. Of those 3 billion base pairs, less than 2% of them actually give instructions for making proteins. That's right, less than 2%. So what's the rest? Well, a huge chunk is non-coding, with a lot of it being these repetitive sequences. For years, scientists called it junk DNA, but guess what we're finding out now? A lot of it plays a super important role in, you guessed it, gene regulation. So that leads to a pretty obvious question, right? If we're all 99.9% the same, how can we tell people apart using their DNA? Well, that brings us right back to that tiny 0.1% of difference. This is what makes a powerful technique called DNA fingerprinting possible. It's how we find your one-of-a-kind genetic barcode. The secret is in these things called VNTRs. That stands for variable number of tandem repeats. Basically, in those non-coding parts of our DNA, there are these short sequences that just repeat over and over again. Now, the key word here is variable. The number of times a little chunk of DNA repeats is different for almost everyone. A scientist named Alec Jeffreys figured out that this variation, which has a fancy name, polymorphism, creates a unique pattern for every single person. And that's it. That's your DNA barcode. So how do we actually see this barcode? Well, it's a pretty methodical process. First, step 1, you have to isolate the DNA from a sample. Then, step 2, you digest it. You use these things called restriction enzymes. They're like molecular scissors to chop up the DNA. Step 3 is separation. We sort all those DNA fragments by size using a method called gel electrophoresis. Step 4 is hybridization, where we use a special probe that's designed to stick only to those VNTR sequences. And finally, step 5 is visualization. We use autoradiography to make the pattern of bands visible. And there it is, the DNA fingerprint. This is exactly what they use in crime scene investigations and for paternity tests. All right, team, let's switch gears. We've got the concepts down. Now it's time to focus on what really matters, getting those marks in your CUET exam. This next part is all about making sure this stuff sticks in your head so you can pull it out instantly when you're under pressure. Okay, let's lock this information in with some easy memory tricks. First, for the Lac Operon, just remember, it's on only when lactose is around. See? Simple. Next, for the inducer, think the inducer inactivates the repressor. The two I's go together. For the Human Genome Project, remember the 3 B's, 3 billion base pairs. And for DNA fingerprinting, just remember what the letters stand for. Variable number tandem repeats. Seriously, pause and write these down. They'll save you in the exam. Okay, practice time. Here's your first question. In the absence of lactose, the repressor, what? Go ahead and pause here. Think about it. All right, you got it? The correct answer is D. It binds to the operator. Why? Because when there's no lactose, there's nothing to stop the repressor. Its default job is to sit on the operator and keep that gene turned off. All the other options, A, B, and C, that's what happens when lactose is present. Question number 2. This is a straight-up memory question, the kind CUET loves to put in. The human genome contains approximately. Pause and lock in your answer. Okay, the answer is C, 3 billion base pairs. Remember our memory trick? 3 billion base pairs. Watch out for those other options, they're just there to confuse you. Okay, let's tackle an assertion reason question. First, look at the assertion. DNA fingerprinting is based on polymorphism. Is that true? Yep, absolutely. Now the reason. VNTR regions vary among individuals. Is that true? Yes, it is. So now for the final most important step. Does the reason explain the assertion? Yes, it does. The fact that VNTRs vary is the polymorphism we use. So the right answer here is that both statements are correct and the reason perfectly explains the assertion. Okay, let's get serious for a second. I want to talk about the common mistakes I see students make on this topic year after year. Please don't fall for these. First, confusing the inducer and the operator. We already hammered that one home. Second, forgetting that the Lac Operon is an inducible system, meaning it's normally off. Third, confusing a VNTR with some kind of gene mutation. It's not a mutation, it's just a natural variation. And finally, that number, getting 3 billion mixed up with 30 billion. Please watch out for these traps. All right, final revision, lightning round. Are you ready? Let's go. Number 1, Lac Operon is inducible, meaning it's normally off. 2, the inducer inactivates the repressor. 3, the Human Genome Project sequenced 3 billion base pairs. 4. Humans are 99.9% genetically the same. And 5. DNA fingerprinting is all about VNTR polymorphism. Boom, that's the entire topic in 5 key points. And now I want to leave you with what I think is one of the most likely conceptual questions you could get on this topic. If you can answer this one clearly, you've got it. The question is, why is the Lac Operon called an inducible operon? Really think about that for a second. And here's the perfect answer you should be able to give. It's called an inducible operon because its normal default state is off. It only gets turned on, or induced, when the inducer, lactose, is present to activate it. That's it. That's the whole idea. Hey, if this explainer helped you get a handle on gene regulation, make sure you subscribe and join a whole community of students who are serious about crushing the CUET. Drop your practice score in the comments, and I'll see you in the next explainer.